So, welcome back to the virtual classroom. Today, I am going to be talking about gene structure as a follow-up to last video about DNA. So we're going to look specifically at the structures of eukaryotic and prokaryotic genes. And they are fairly different from one another due primarily to the chromosome structure that is different between each type of organism. On the whiteboard here, I have a set of models laid out that represent a eukaryotic cell. Right? So we have this big blue circle is our eukaryotic cell. We can tell that it is eukaryotic because it has a nucleus, which is represented here in the middle. Inside the middle, we have these squiggly lines, which when we zoom in, we see our DNA molecules, our chromosomes. Right? And so these are in their functional state. They are not the tightly compacted meiotic or mitotic chromosomes that we usually think of. These are the spread out, ready to be used chromosomes that are functional for genetic expression. If you recall from the last video, these chromosomes are lined with circular ball-shaped proteins called histones that give them their bends and regulate how tightly coiled they are. And they are anchored to the nuclear envelope in certain specific spaces. So they have little physical addresses where you can always find them. This particular organism I have decided to model with 3 separate chromosomes. So its entire genome would be 3 chromosomes large. I've only given them 1 copy of each, so this would be a haploid organism. Okay? So this is a haploid organism with 3 distinct chromosomes. Zooming in on this particular region of this chromosome, we have a particular area that I've identified as a gene, which is, of course, today's topic. So this is the level of conversation that we are on. We are looking at a very specific region in a very specific chromosome. So that is what we are exploring, and I will be right back with a fresh and different model. Okay, so before we start exploring the actual structure of a gene, I thought we could use a little refresher on what specifically a gene is. I know you're all familiar with the general concept, but just a little reminder from Biology 1. A gene is a specific region of a specific chromosome that codes for a specific trait. So this region within a species will always code for the same general trait, be it eye color, enzyme activity, height characteristics. Any trait that is genetic is controlled for by at least 1 gene. In reality, most traits are a combination of multiple genes, which is something we'll explore when we get to inheritance patterns. But as a general definition, this gene is a specific region on this chromosome that codes for a specific trait. Now, these regions, if you recall from the last video, are thousands of base pairs long. The longest is like 220,000. Right? The typical one was around 67,000 base pairs long. So obviously, I'm not going to draw out 67,000 base pairs. So this is the model you get, okay? And you don't get upset. So this gene is always in the same space, but the specific sequence of nucleotides that show up within that space can vary. And that is where we get genetic variation of traits. Right? So the difference between brown eyes and blue eyes would be that brown eyes has this sequence, and maybe blue eyes trades this CG out for a TA. Right? And maybe having this here breaks my gene so it doesn't work properly anymore. The shape of the protein that this codes for isn't going to do the same job. So that variation is called an allele. Okay? And so alleles are these variations in nucleotide sequences. They are not the variations in the traits. You can have different alleles and still have the same presentation of that trait. If I make this change here, this is still a different allele, right? But maybe they both code for brown eyes. It's possible. Make sense? Cool. So that is our general refresher on what a gene is and also what alleles are. I will be right back with a more specific view of the structure of a gene. Okay, so here we have a very clear model of our gene structure for eukaryotic organisms. We've zoomed out away from the DNA so you can no longer see the specific base pairings. Because as I said before, this is covering, 67,000 to 200,000 base pairs. So in order to see the whole gene, we can't be that zoomed in. So we've zoomed out and we're looking at the DNA strand. Okay? The gene itself is this region here that I've highlighted with a bunch of boxes. Only one side of the DNA is coding for this gene. And so that side in this particular model is running from 5' here to the 3' here. Okay? And the direction is important. And so we have the upstream area, we have the downstream area. The gene is going to be expressed first through transcription. Right? And so we need transcription to occur, and that's going to happen with a molecule called RNA polymerase. Sorry, my computer is being jumpy, so excuse the messy writing. You'll have to imagine that this is clear. It says RNA polymerase, right, which let's just make it a ball-shaped enzyme. And it needs to attach to the gene in the appropriate place. The place where the RNA polymerase can attach is called the promoter. So this front region here at the upstream side of the actual gene is called the promoter. There are some sub areas of the promoter as well, one of which is called the proximal control elements, and the other is the Tata box. Okay? So these things in general, this whole area is where the RNA polymerase can attach. Now this gene can be turned on or turned off by modifying this region. If we change the shape of this region so that the polymerase cannot attach, this gene is off and will not be expressed. If we change this region so the RNA polymerase can attach, then this gene is on and will be expressed. And that's what's going on with these control elements. These control elements are responsible for regulating whether or not this area is going to be shaped appropriately for RNA polymerase to attach. Okay? So we have the promoter to engage with RNA polymerase. After the promoter, we have the actual coding region of the gene. Okay? And so for eukaryotes, you'll have one structural gene here. This area will code for one protein, effectively, in most cases. Okay? So you turn this on, it runs transcription, makes RNA. That RNA then goes out, meets up with a ribosome to generate a protein through translation. That's what this gene does here. Okay? So this is the gene region. Way upstream from the gene itself are the distal control elements. And they, like the proximal control elements, play a very important role in modifying the shape of this DNA so that it can be activated. There are secondary molecules involved in this storyline. They're effectively ligands, right? Or the product of ligands, rather, because ligands are the communication that happen outside. But these can be the products that happen through cell communication as one of the options. Okay. These molecules are called transcription factors. If my computer wasn't being so sketchy, I would write it all out, but often you'll see me abbreviate it as TF anyways. So these transcription factors engage with the control elements. And at first it engages with the distal control element, turns the whole thing on. There's a series of steps that ultimately bends up this whole DNA strand so that the distal control elements end up above the proximal control elements. That allows for a cascade of reactions that bring in positive transcription factors that will engage with the proximal control elements, and that engagement is what allows the RNA polymerase to fit onto the promoter. Okay. When we look at the regulation mechanisms, we will go over these steps in detail about what it is that I'm talking about right now. But for now, you just need to know this general outline of the gene structure. Okay, so we have the gene itself. The gene is composed of the promoter region, which in and of itself is composed of the proximal control elements and the TATA box. And then after that, downstream, we have the actual coding region, the structural gene itself. That's going to code for the protein. Way upstream from the gene is the distal control elements that play a role in regulating whether or not this gene is on or off. Okay, so that's the gene structure for eukaryotic organisms. All right, so onwards to prokaryotic gene structure. Now you got to keep in mind that the prokaryotic chromosome is a single circular chromosome, as opposed to the multiple linear chromosomes of eukaryotic organisms. The single circular chromosome has its benefits and its downsides, and one of the benefits is the way in which prokaryotic organisms can efficiently organize their genes. Because they're all on one singular chromosome, evolution has led to the development of what are called operons. So within the genome of the prokaryotic organism, there are these collective gene regions called operons. So instead of just being a singular gene here, there are a bunch of related genes all under the same regulation mechanism. Right? And so if this was the cheeseburger gene, one of these genes might be the bun gene, one of these genes might be the beef patty genes, and one of these genes might be the ketchup gene. Right? And so if I want my cheeseburger, I want to turn on all those genes at the same time. The operon allows for this uniform regulation. Very efficient, very cool stuff. Okay. So we are looking at the actual operon here in this region. Again, we are dealing with a 5', 3' direction. So we're downstream here and upstream here. Okay. And within the gene itself, in the operon itself, rather, we again see a promoter region, and the promoter region serves a similar function to the promoter region in the eukaryotic organism. This is where the RNA polymerase can attach to the gene when it needs to be transcribed. Okay? Unlike the eukaryotic gene, we see a unique region called the operator within the promoter. Okay. And the operator is very important to regulating whether or not this operon is functional or if it's dormant. Okay. And so this region is shaped in a way that allows for the attachment of a secondary molecule. And this secondary molecule either blocks the progress of RNA polymerase when it's there, or if I remove it, RNA polymerase can roll on down and transcribe these genes. Okay. So the operator is where that molecule is attaching, and that is blocking or not blocking the RNA polymerase. Upstream a bit from the operon is a regulatory gene region. This is not technically part of the operon itself, okay, but this region codes for the molecule that fits onto this specific operator. So this molecule is made by this gene, okay? And if the operator is active, it fits in, boom, 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 blocks transcription. If this is not active, this doesn't fit in, it just floats away, goodbye, and RNA polymerase can do transcription. This is another thing that we will look at more specifically when we're talking about gene regulation mechanisms. Okay? But for now, we're just looking at the general structure of the gene. So we have our operon, right, which is composed of the promoter, which includes the operator, and then we have the related structural genes that all have some kind of similar function to one another. We want to turn this on or turn this off altogether. Okay? And then upstream we have the regulatory gene region that codes for this regulatory molecule. And that's it for now. Cool. Awesome. So that's it. That's all I wanted to talk about here in today's little video. Just wanted to show you the structural aspects, effectively the anatomy of the genes of eukaryotic and prokaryotic organisms. We will explore these very fascinating, very cool, very fun mechanisms at depth during the regulatory mechanisms unit area topic time. And I can't wait because I love it. I love this particular unit. I love all biology, really. I feel like every unit, I'm just like, I love this. I don't like macromolecules, though. I hate macromolecules. That bores me. But the rest of it's fun. So, yeah, that's it. Have a great day. Bye.